Hello and welcome to BioCode. In this video, we are going to have a thorough discussion about single-cell RNA sequencing technologies. There are various different technologies in single-cell RNA sequencing, and we do have the opportunity to choose from multiple different technologies. Each of them vary in terms of their cell lysis, cell isolation, how the cells are taken from the tissue, and how they are further sequenced, how they are prepared for a library preparation. So back in 2009, Tang et al. Started working with manual labor where they were able to manually lyse the cell from the tissue, and they were able to manually prepare cells for single cell sequencing. This was a really early, back in even really early for third generation, next generation sequencing procedures. So from there on, multiple thousands of different researchers all around the world started developing these different technologies that we can see over here. STR-seq, Smart-Seq, CEL-Seq, and Drop-seq, in Drop-seq, 10x Genomics. These are the most crucial technologies that are still utilized these days, but they were developed like almost a decade ago. So almost all these technologies vary in terms of their procedures of development or procedures of library preparation and how the cells were obtained. Other than that, the analysis of the data generated from these technologies does not vary that much. The analysis procedures and the analysis steps are almost similar. The only difference is how the cells were obtained, how they were prepared for sequencing library, how the RNAs were extracted from them. That's it. Single-cell RNA sequencing has outgrown the other technologies, especially in genomics work, and it is highly utilized technology and even has developed further technologies such as spatial genomics and spatial transcriptomics. So there are two broad categories of single-cell RNA sequencing technologies. First one is full-length transcript coverage, and the other one is 3' and 5' coverage. Both of these technologies help us to either do full-length read sequencing or do 3' or 5' of the end of each read and do sequencing of all those ends only. So what does it mean? It means that in 3' or 5' end, we are only sequencing the read or the fragment of the RNA or the cDNA from its 3' end or the 5' end. The technologies that do this kind of work is Drop-seq, Seq-Well, Seq, STR-seq, 10x Genomics, and other Drop-seq technologies. These are also known as droplet technologies. There is going to be a proper video on 3' and 5' end coverage sequencing technologies, so don't worry about it if you don't understand them thoroughly yet. On the other hand, full-length transcript helps us to do sequencing of the entire transcript rather than the 3' or 5' end coverage. So for full-length transcript, there are three core mostly utilized popular techniques such as Smart-Seq or Smart-Seq 2, MATQ, Super-Seq technologies. The difference between them is how the data was obtained and how the data was prepared for sequencing and what sort of fragments were sequenced, either full-length transcript or 3' or 5' end only. So which one to choose from, one or the other? Each technology has its own distinct feature and drawback. Choice of the technology for the experiment comes down to the research goal. So it all depends upon the research goal, what you are trying to do. We will discuss them further. The depth of the sequencing is shallow, often depicts only 10 to 50% of the transcriptome per cell. So no matter which particular technology you use, single cell is still not the best in terms of detecting like 100% of the transcriptome per cell. It can only do 10% to 50%, which is an issue. Even though we have huge data set, like thousands of cells, but we still don't have proper sequencing depth. We will have a discussion about sequencing depth further. So what is the difference between these technologies? Why do they exist? So each technology comes by time, and if we go back, we can see that when single cell started developing, there were multiple different technologies that were based on the previous methodologies, or they were innovating something. And if we come down to here, there are 5 major steps in each of this technology. No matter what single cell technology it is going to have these 5 steps. The first one is cell isolation. how the cell was isolated from its sample, from its tissue, from the original sample. Second step is reverse transcription. Third step is second strand synthesis. Fourth step is cDNA amplification, and fifth is library construction. So among all these technologies, either full length or drop-seq technologies, 3' or 5' end, these 5 steps are similar in terms of these are the steps and this is the order in which these steps are going to be followed. Either and what they do is different among all these technologies, and we are going to discuss them. So if you see over here, Smart-Seq 2, MAR-Seq, scRB-Seq, all these 3 technologies are utilizing fax sorted machines for sorting the cells based on its condition for further lysis, cell isolation. Drop-seq utilizes microfluidics. CEL-Seq, Smart-Seq original, which was C1, also utilized a type of microfluidics technology. Drop-seq utilizes the droplet technology, basically, that is part of microfluidics in some sense. We are going to have a thorough discussion about these topics further as well. So reverse transcription, no matter how the cell is isolated, it has to be reverse transcripted using reverse transcription procedures. And in this step, what happens is that we attach UMIs or unique molecular identifiers, barcodes of the cells, to these particular fragments that we have obtained through cell isolation and library preparation. And if you see over here, Smart-Seq original and Smart-Seq 2 takes the read and attaches PCR adapters, whereas scRB-Seq has UMI, barcode, cell barcode, and PCR attached to it as well. MAR-Seq also has UMI, barcode, and T7 adapter. Drop-seq technologies such as Drop-seq, UMI, barcode, and PCR adapter. Similarly, CEL-Seq. So there is not that major difference. The only major difference is that Drop-seq technologies utilize UMIs, which are unique molecular identifiers that allow us to attach a set of synthetic DNA or set of synthetic fragment to each read or each transcript obtained from each given cell, so that we can tag it using a unique molecular identifier, and we can identify it after sequencing. A cellular barcode is also attached, so each cell has its own cell barcode. So no matter how many cells it has, our sample has, each cell type will have its own cellular barcode. This allows us to identify cells after sequencing and allows us to identify transcripts after sequencing. The reason for that we will discuss further. So no matter what we attach with our transcription, either barcode, UMI, or PCR adapter, we do have to do second strand synthesis. This is where we prepare the second strand. And after second strand, what we do is we do cDNA amplification. We have to do PCR. Some technologies, CEL-Seq and MAR-Seq, utilize IVT procedure, whereas other technologies that are most commonly utilized technologies do PCR amplification. And this is where library is amplified. For example, we had one fragment, we will now amplify it to 100 times so that we have more depth in our sequencing data, so that the sequencing procedures are proper. We remove these overly duplicated information later on, and this is where UMIs and cellular barcodes help us to identify technical duplicates that we have created ourselves using PCR, or the original duplicates that might be possible due to overexpression of a particular gene. After this, tegmentation and 3' enrichment and other procedures are done to do library construction. So the major difference between all of them is whether or not this technology utilizes UMI or not. So CEL-Seq utilizes full length, it utilizes UMI, but it is not full length, it is 3' or 5' end. Drop-seq utilizes UMI, not full length. Smart-Seq, UMI, not full length. scRB-seq, scRB-seq, UMI does not do full length transcriptome analysis, and Smart-Seq does not have UMI. Smart-Seq 2 does not have UMI, but it allows us to do full length transcription. So the discussion that we were having earlier, that either it is going to be full length or 3' primer, and this is where it gives us further knowledge. So what is the difference? The difference could be due to cell isolation, cell lysis, reverse transcription, amplification, transcript coverage. Either it is full length, 3' and 5' and strand specificity, either it is positive strand, negative strand, either we are attaching unique molecular identifiers or not, how the cell is isolated, how the cell is lysed, how the reverse transcription is performed, either or not, whether or not amplification was done or not. So let's visualize the difference further. Imagine that we have cells, we have taken a tissue and we utilize FACS sorter, which is a basically machine. We will discuss how the lysis is done in the further videos. So this machine basically is able to utilize laser mechanism to identify and sort the cells into multiple different populations of cells. As you can see, physical separation of cells into 60 well plates. So each cell goes into its own well. We do have fluidium C1, which basically allows the physical separation of cells using microfluidic chip that I was discussing earlier. So these three are different main technologies. These are known as gel beads, FACS sorter, and fluidium C1. So fluidium C1 allows us to do physical separation of the cells on a microfluidic chip. It utilizes Smart-Seq 2, Smart-Seq, STR-Seq. These are the technologies that utilize fluidium C1. Whereas FACS sorter, the cells from this sorting go to Smart-Seq or from over here as well. Then from over here, FACS sorting also utilizes CEL-Seq, MAR-Seq, scRB-Seq or CEL-Seq. Whereas Drop-seq, in Drop, 10x Genomics, all of them utilize these particular beads where the cells are trapped inside hydrogel droplets and then they are further pooled. Then amplification method is also different. How the amplification is done, pooled PCR amplification or individual cell amplification. Further, sequencing method is also different between them. We are either doing 3' sequencing, just this highlighted region is sequenced for each transcript, or we can do full length transcript analysis. So this allows us to do detection of gene expression. If we just want to identify gene expression, if we just care about gene expression, subpopulations, that's it. Then we just need to do 3' or 5' and sequencing, not the full length. But if you need to do gene expression analysis, splice variant, isoform switching, alternate splicing, BCR, TCR, diversity analysis, then you need to have full length sequencing available with you. So the choice of transcript sequencing technology depends largely on what exactly do you want to do. If you are looking for 3' or if you just want to identify the best results and you need to have cheapest possible cost of your entire analysis, and you just want to identify cell populations, cell type populations, you just want to identify gene expression analysis, then you need to do 3' or 5' and sequencing. That's it, because it allows us to do more accurate quantification using the use of unique molecular identifiers, which we have discussed previously. It allows us to distinguish biological duplicates from amplification of PCR duplicates. So basically what happens is that UMIs are attached to multiple different transcripts. So if each read comes from the same transcript, it will have same unique molecular identifier. So it allows us to identify those biological duplicates and allows us to do amplification as well, so that we can later on remove these PCR duplicates. It provides best results with more than 10,000 cells. Full-length sequencing, on the other hand, as we can see over here, Smart-Seq technology, the cells are sorted using FACS sorted. Then we have the sequence and the entire transcript is sequenced, double strand is created, bead purification, validation, and then sequencing library is prepared and sequencing is done on the full length transcript. It allows us to detect isoform level differences in the expression. It allows us to do identification of allele specific differences in the expression. Deeper sequencing, a smaller number of cells can also be identified. Best for samples with low number of cells is possible. If you have low number of cells, but you are doing full length sequencing, the results are going to be very good. But if you have higher number of cells, then you can do 3' and 5' and get almost similar results in terms of gene expression analysis. For example, Smart-Seq 2 can find bigger number of true expressive genes than other technologies such as CEL-Seq, Smart-Seq, MAR-Seq, etc. Because we are doing full length transcription and the sequencing depth is more as compared to the other technologies. MATQ-Seq can find genes with low abundance better as compared to the others. Both can capture poly A or regions. But the fact is that we need to understand what exactly we are trying to do. Either we are trying to do cell clustering, cell population analysis, tumor heterogeneity analysis, gene expression analysis. Then drop-seq technologies are fine with us. We can utilize them. But if you want to do further in-depth analysis, diversity analysis, TCR, BCR, diversity analysis, isoform switching, etc., then you need to do full-length transcript sequencing. So difference between the full-length and 3' or 5' and transcript coverage is that full-length gives us more transcript coverage, allows us to do allele expression, RNA editing, isoform switching. It gives us low abundance or lowly expressed genes in our analysis as well. For example, those genes that don't have proper expression in the cells, they are very low. They have very low abundance in nature. Even those can be identified using full length transcript analysis. But whereas on the other hand, 3' and 5' and transcript coverage